The seminar is to be to have a presentation by Ralf Schwamborn. I hope I'm pronouncing his name correctly. He is an associate professor from the University of Pernambuco in Recife, Brazil. And his area of specialization is marine biology and fishery biology. And he's currently teaching at the same university. He's currently teaching by basic statistics, trophic ecology, method of food webs, and modeling and advanced statistics using R. The topic for today is, as indicated on the schedule that was circulated, which focuses on the biomass spectra and stable isotopes in oligotrophic and upwelling ecosystem of the tropical and sub-Antarctic consequences for the ecosystem predictions. Without wasting time, I think I will allow the presenter to start with the presentation. Ralf, you can start with the presentation.
Okay, thank you very much, Johannes. And first of all, I would like to thank you for the invitation, also to Margit Wilhelm, who's also present for the invitation, and also to Kurt Hanselmann also for the invitation to this seminar. And also I'm very happy to see some familiar faces here. For example, I saw Henrike also here at the audience, and other familiar faces, so I'm very happy to be here. It's a privilege to be here and to present some recent results regarding biomass spectra and stable isotopes in the tropical and South Atlantic, and talk a little bit about the consequences for ecosystem prediction in the context of the Triatlas project. So I'm just going to start right away. And first, it's important to remember that marine food webs are highly complex. So complexity is the main problem that we face, when we try to predict the future of marine ecosystems, the high biodiversity. For example, when I talk to physical oceanographers and many other people who are not familiar with ecology and biology, they always get really overwhelmed when we talk about the numbers of species and forms and sizes and feeding strategies of the organisms. And to make things even more complicated, it's highly dynamic in time and space. So the oceans are very difficult to predict when we talk about ecosystems. So how can we do the prediction? So there are many different approaches to this. The current approaches that we see, they range from the most simple models to highly complex individual-based and spatially resolved models. I like to do very simple things. So what is the most extreme simplification is to forget about all, let's say, complicating factors and look only at the level of the atoms of, for example, carbon and nitrogen atoms. And then we can distinguish them, for example, by stable isotopes. For example, what I'm showing here, you can see here is the stable isotopes of carbon, carbon 13 and carbon 12. And another way to simplify such complex ecosystems is looking only at the size of the organisms, which would be the context of size spectra theory, where we look at the size distribution and the abundance of the different organisms. And we will see today how we can combine these two approaches to understand a little bit more about marine food webs and maybe also some useful aspects for ecosystem prediction. So what are stable isotopes? I'm going to be very quick here. Most of you already have an idea what stable isotopes are about. First, Stable means that they are non-radioactive, and it's opposite to radioactive isotopes, for example, C14, which you know is radioactive. So the isotopes, what does it mean? Isotopes are elements that have the same number of protons, but more or less neutrons. That means they have only a difference in mass, no, have the same chemical properties, but different mass. For example, here, these are some isotopes of carbon, where the C11 and C14 are radioactive, and C12 and C13 are not. That's why we call them stable isotopes. They can be used as tracers in food webs. The other aspect that we are approaching here are size spectra, which is a way to show ecosystems looking at the way that the abundance decreases with body size. And usually people assume and often also observe that there is a log linear decrease in abundance with body size. Here you see, for example, the high abundance of very small organisms like phytoplankton, and then it decreases a lot when we go to zooplankton, and then they're comparing to the former organisms, the phytoplankton and the zooplankton, there are very few fish per volume unit. So you have less of the larger organisms, and the shape of this decrease is what we call the size spectrum. This has been developed as a theory and a method since the 70s, so we already have a very strong theoretical background here. So, and what determines the shape of the size spectrum? First of all, we have to define shape, and one of the main aspects of the size spectrum is the slope. That means how steep this slope is. No, we can have a very steep slope or a shallow slope of the log linear size spectrum. And what defines the slope? So there have been lots of publications regarding the, let's say, the models that can describe or predict the size spectrum slope. Some have focused more on the trophic efficiency. There are many papers that focus on trophic efficiency, like the ones that we have also published recently, like which is then the energy and mass transferred from a trophic level to the next one. Some other papers have focused on the predator-prey mass ratio, which would be the mean ratio of body mass in the food web. And then we also have the metabolic scaling, which is the change in metabolism with size. And all of these can be influenced by several processes and parameters in the ecosystem. For example, the density and availability of food, temperature and oxygen, and some other parameters. And just simplifying a little bit, we can say that the slope, the log linear slope of the size spectrum is approximately proportional to the log of the trophic efficiency to the log of the predator-prey mass ratio. So this ratio of trophic efficiency and predator-prey mass ratio can define the slope. And if we assume a metabolic scaling of approximately 0.75, then we can get this nice and simple equation that we can use to predict a size spectrum, assuming that we are using the so-called NNSS size spectrum, which is the normalized numbers size spectrum. It's a way to assemble the data, and then we get this, in theory, this nice equation. And we will have a look at how we can use our data, our size spectra data and the stable isotope data to fill in some parts in this equation and possibly obtain estimates of trophic efficiency in the ecosystem. So how are we doing this? One of the central methods in our working group to study marine ecosystems is the ZooScan, which is a simple apparatus like a scanner, like the scanner that you use to scan documents. but that has been modified so it can receive wet samples. And this apparatus then gives us a scanned image of a sample. Now this here is an example of a zooplankton sample with these beautiful little zooplankton organisms, and with such a digitized image we can describe the size and also make an estimate of the volume of the organisms using a three-dimensional ellipsoid model, no, which is the ellipsoid volume. Also, we can use these digitized images for semi-automatic identification of the organisms, where we actually do also manual validation always, no, because the algorithms are not always 100% reliable. So the identification is still verified. We have a semi-automatic step with algorithms, and then we have human specialists and taxonomists who do the verification of the algorithms for the identification. But the measurement and the volume calculations, they are done automatically with algorithms. Are you following me?
Yes.
Yes.
Thank you very much. So let's go to a few recent results. For example, using this ZooScan method, we have described the composition of the plankton and particles in tropical coastal ecosystems. And to our surprise, we found out that particles are not only very important and abundant, but they can be as abundant and as important in terms of biovolume as the zooplankton, and in some samples even be more abundant and more total biovolume than zooplankton. So we don't have only zooplankton in our samples, we also have a lot of non-organism detritus particles, are very important also. You know, this here was our work with Natalia Lynch-Silva, who also has recently finished her PhD thesis here in our lab, with Katarina Marcolin from Bahia State. This is one of the central results, no, where you have these black bars, zooplankton abundance or volume, and the gray bars are the particles. So you can see there are lots of particles in the system too, and it's also very interesting to do the, let's say, the analysis of the size spectrum and composition also of the particles, not only of the zooplankton. So, and within the particles, we have been characterizing also the microplastics with help with a working group in South Brazil from FURG, and microplastics are also very abundant and also important in volume, and we're also describing the size spectrum of the microplastics. Another PhD thesis that was completed recently in our lab was the one of Gabi Figueiredo in the context of the Brazilian-French Abraços cruise and research cooperation project in the shelf, offshore and oceanic island areas of northeastern Brazil on board of the French research vessel Antea. And one of the main focuses of her PhD thesis was the stable isotopes and also the size fractioned stable isotopes, where we did the analysis of the different zooplankton samples after sieving them into size fractions. So we have a relationship between the stable isotope composition and the size, and with the nitrogen isotopes we can estimate the trophic level, and we can use the carbon isotopes to estimate primary carbon source. And these dark symbols here are the POM, the particulate organic matter. And you can see that the particulate organic matter is quite different from the zooplankton, which probably is due to selective feeding. And also you can see the different colors that differentiate the different size fractions. And you can see that the size fractions have different stable isotope signatures. And this is regarding the nitrogen means that they have most likely different trophic levels. So we used these data to calculate the trophic level by size class. And we have here on the x-axis the different size fractions, which will be the mean size in each size fraction. So on the x-axis you have the size, and on the y-axis you have the trophic level that was calculated from the nitrogen isotopes. And you can see that there is a very nice increase in trophic level with size. So this proves that there is a clear relationship between size and trophic level in these zooplankton samples. And this slope that you see here, this dotted line, can be used to estimate one of the components of the equation that I showed previously. We can use this slope of the trophic level size slope. We can use this to estimate the predator-prey size ratio. This slope is intrinsically connected to the way that size and trophic level are related. And sorry for that, just turned it off here. And are you following me?
Yes, all good.
Very good. So, and we can then connect the predator-prey size ratio with this slope here for this equation. So we can estimate the predator-prey size ratio. And also we calculated, of course, the size spectrum slope for the different regions, for example, for the shelf region and the oceanic islands. So this will be the NNSS slope, the normalized numbers size spectrum slope, where normalized numbers means we have here the abundance. No, we can also have the biomass or biovolume size spectrum. Here we are talking about abundance. That means how the numbers of organisms decline with size or mass, or in this case, volume. Well, this here are the bigger organisms that have a larger individual biovolume, for example, large jellyfish, and here you would have a very small copepods, small crustaceans. No, and this is the individual biovolume on the x-axis, and on the y-axis it is the abundance. So the very small ones are very abundant, and then we have a nice log linear decrease in abundance with biovolume. So how do we combine these data? This was also part of Gabi Figueiredo's PhD thesis. We used the nitrogen isotopes to calculate the mean predator-prey mass ratio. We put this into this equation here, and then we transformed the equation. And here we have the trophic efficiency. So the trophic efficiency can be calculated as the predator-prey mass ratio, and here is the NNSS slope, and here would be the metabolic scaling. And one thing that is very interesting here is to see that we got for the first time such estimates for these areas. And the other interesting thing is that the way that we calculate the trophic level from the stable isotopes is a key source of uncertainty here. So the transformation of the data from stable isotopes to trophic level is far from trivial, because, for example, we don't know the trophic fractionation very well. So this was also one of the key insights from this PhD thesis and the papers that we published from this thesis. Here again, coming back to the work of Natalia, we also have these biogenic particles and microplastics. And here the study areas, in the Rio Formoso estuary, the Mandarebe and the continental shelf. We see that there's a difference in salinity. The estuary have lower salinity, and also the second depth we have very turbid estuary. And we have the size spectra here of zooplankton, detritus, and microplastics, the different areas. And also we got the size spectrum slope for each sample, where we have calculated the slope for zooplankton only, zooplankton with all particles and everything, and also without microplastics. So we also simulated a world without microplastics to compare these three models. No, and maybe most interesting and most relevant in this context was the recent comparison that we made in the Triatlas project, which is still ongoing, which was, let's say, shared within the Triatlas deliverables, which is the comparison between northeastern Brazil, which is a very oligotrophic area, and the Benguela current ecosystem, which is a very productive upwelling system. No, this was so our first deliverable in the context of the Triatlas project, which is funded by the European Union, and several people who are attending here are also part of the Triatlas project. No, we combined several institutions from Europe, from Africa, and also from Brazil, including also from Namibia. And this was this deliverable, which was a report on food web metrics and physical, chemical, and biological process rates, where together with Heino Fock, myself, we were the coordinators of this synthesis effort, and I'm just going to present some, let's say, preliminary results from this report. And many people contributed to this report. It was a very big effort, a huge effort with many, many people who contributed to this. And the study area here in Triatlas is then the tropical and South Atlantic, and here we have the Benguela current system, and here we have the oligotrophic waters of northeastern Brazil, where we have the North Brazil undercurrent and the South Equatorial current, which brings oligotrophic warm waters here in the direction to Brazil, and is responsible for these oligotrophic conditions with a permanent thermocline, which is very different, an extremely different system from the Benguela upwelling system, that most of you know very well. And the idea was to explore and compare size spectra across different ecosystems, and investigate productivity and trophic transfer, especially to having a combination of zooplankton and fish, which Tim Dudek and Henrike Andersen, they are mostly responsible for doing this, together also with many colleagues also here from Brazil, like Simone Lira also, and Gabi Figueiredo, and many colleagues also from the Federal Rural University, a lot of people, and it's a huge effort to combine all these data from zooplankton and fish from these areas. Very large ongoing effort. We are still far from finishing this, and I'm going to present some preliminary results that we obtained here in the context of the Triatlas project. One important result was from the UVP data, the underwater vision profiler data, which is an equipment that was developed in France, in Villefranche-sur-Mer, and it was deployed in many, many places in the world. And clearly, if you see here these steep slopes, which are the dark pixels, and the shallow slopes, which are the yellowish green pixels, you can see that areas that are shallow, that are very productive, they have shallower slopes. When you compare, for example, here off Namibia, you have lighter color, no, a light green color. And if you go to the oligotrophic areas here of northeastern Brazil, we have a darker bluer color. So you have shallower slopes in productive areas, and you have a steep slope in oligotrophic areas. You see the dark pixels here means steep slope, which means probably less efficient systems. That indicates that where the system is less productive, it is probably also less efficient. Also, we did a lot of work comparing the size spectra, demersal and midwater trawls, including fish, also historical and recent data, and also with the mesopelagic macrocrustacean communities, where here we also obtained the size spectra for these macrocrustacean communities for the first time. And the methods designed comparing northeastern Brazil and the Benguela upwelling system, the different cruises and the different, let's say, approaches were quite comparable, which allowed us to do these nice comparisons of these different systems, where we have the North Benguela upwelling system, the South Benguela upwelling system, offshore and shelf, and we have the three Northeast Brazilian oligotrophic systems, where the oligotrophic systems are in blue, because it's blue water, no, it's oligotrophic, and green water Benguela upwelling system here. And you can see that the abundance, the zooplankton, is much higher in the Benguela upwelling, and also the wet biomass in the Benguela upwelling system is much higher than in the oligotrophic systems. And also on the shelf, it's much higher than offshore. No? You can see the highest abundance and the highest biomass was found in the North Benguela upwelling shelf of Namibia. That's where we had the highest abundance and the highest biomass of zooplankton in this project. So then to define and combine these different data was a huge challenge, and it's still a huge challenge to define the starting and finishing classes and the gaps. And so it's really it's a huge effort and there are lots of very difficult decisions to be made. No, these are some examples. This graph was made by Tim Dudek recently, and we see here that there is a very nice log linear decline also in system abundance per size class when we look at the individual biomass. No, these are the large organisms which have a large individual biomass. These are the smaller organisms which have a small individual biomass. And this is the biomass within each size class. And you see there's a nice decline. And then we compared these slopes. It was also made by Tim Dudek recently, comparing these slopes of the different systems. And when we look then at the NBSS slope, which is the normalized biomass, or in this case, biovolume size spectral slope in relation to the biomass, then we can see that the North Benguela upwelling system shelf of Namibia has the highest biomass and also has a very shallow slope. That means that it's very efficient compared to the less efficient tropical system, which are these blue dots here. That means that if we look at a global warming scenario where probably there will be an increase in temperature and in surface waters, and also an increase in mixed layer depth, then we must assume that not only the productivity, the primary productivity of these systems will decline, but also then unfortunately also the efficiency will decline. So most likely there will be a shift from this direction of the graph with higher biomass and higher efficiency towards less efficiency like a tropicalization of the systems towards a less efficient and less productive state. Now, this would be the main consequence for ecosystem prediction from this preliminary analysis. So the main conclusions here would be that steeper spectra in oligotrophic areas, they confirm previous studies. And in some cases, we had some S-shaped shapes, but it's still under debate. We're still debating these things. And large biomasses like krill, they can obscure this image. And macrozooplankton such as krill, salps, and jellyfish are still absent. And we still have these problems with the gelatinous organisms, which were subject to a very nice talk also recently, here in the context of the seminar, the gelatinous plankton. So the general conclusions here also, when we look at the phytoplankton and the zooplankton regarding biomass and the size spectral slope, is that steeper spectra are generally found in oligotrophic systems, and this is something in the context of this Tri-Atlas report that we found for the UVP, these in situ optical system data for the phytoplankton and also for the zooplankton. And so the hypothesis that we are analyzing and probably are going to confirm in the near future is that oligotrophic systems may be probably less productive and probably also less efficient. And the main consequence for ecosystem prediction would then be that this decreased efficiency and decreased primary and then much decreased secondary productivity with warming and deepening of the mixed layer. So we would have a sequence with warming, decreased primary production, and subsequently decreased efficiency, and then a much decreased secondary productivity due to the decreased efficiency and the decreased primary production. So, but still we are far from finishing these analysis. We're just at the beginning of it. We're starting to do such analysis, and there are many problems that we are facing. For example, the effects of temperature and oxygen. Also, we talked a lot about predator-prey mass ratios, but they are still far from resolved. I just talked about the problem from transferring the stable isotope data to these predator-prey mass ratios. And also we don't have very good data from estuaries and nearshore shelf areas. And also mesopelagic fish communities are still very difficult to describe and understand. And we are working on this at the moment. So this is still ongoing work in the context of the Tri-Atlas project, specifically the synthesis working group, where the combination of size spectra and stable isotopes is one of our key approaches in this synthesis working group, and in the different combinations of co-authors and smaller working groups. So thank you to everybody who participated in this, and also thank you very much for your attention.
Well, thank you, Ralf, for a good presentation. And we also have now sufficient time for questions and discussions regarding what he presented. I would prefer that you raise your hand, but if you cannot, you can also write on the chat and I can read it out. I see the first head I see is from Margaret. Margaret, you can say something.
Thank you. Hi, Ralf. Thank you so much for the very interesting presentation. My question is about your comparisons of the slopes of the different systems. And I know you were comparing more the oligotrophic system with the upwelling system. But what is interesting to me is that the northern Benguela and the southern Benguela upwelling system are, or let me ask, are they, if you compare those two and their efficiency to one another, are they, is that a surprise? Was that a surprise? Maybe if you can go back to that slide. That's one of your last slides.
Yes, comparing the different systems.
Yeah. Yeah, so am I correct to understand the top one is the less steep slope, which means it's the more efficient, it's a
yes,
higher
yes.
transfer efficiency.
The top systems are the more efficient ones.
Okay, and is it a surprise to find that the northern Benguela is much more efficient than the southern Benguela?
I understand what I understand where you want to arrive here, but I think we still are working on this, and these are very preliminary results. What means that we are still doing the statistical analysis of that, and I have the impression from the data we still didn't finish this, and we still didn't even start doing proper statistical analysis of that. So this is very preliminary. These are just some first impressions, but my impression is that the difference in efficiency between South and North Benguela upwelling system in these data sets may probably not be so relevant and maybe even not be statistically significant, depending on the results. I'm not sure. We didn't do this yet, no, but I understand why you want to get, because as far as I remember, there's a lot of literature saying that the Northern Benguela system is less efficient, right, Margit?
Yes.
Yes, I know, but I'm not sure. I'm not sure whether we will be able to overthrow this paradigm here. We are rather comparing the extremes. Let's say we are focusing on the comparison of the oligotrophic systems and the upwelling systems. But I don't.
Yes.
I'm not sure whether we will have very strong conclusions that will be enough to compare the Northern Benguela and the Southern Benguela systems, as you are now interested in. I understand that this will be very interesting, and but I think it's not the focus of this project, and I'm not sure whether we will be able to do that. I think the main focus of this at the moment right now is to look at these extremes, the very oligotrophic tropical Atlantic on one side, and looking at all these different large systems like the oceanic islands and the offshore and the shelf on the one side, these 3 blue dots, and looking at the 4 green dots, the 4 upwelling systems, and comparing these 2 data sets. I think that's, if we get this well done in this project, and if we can do this with our friends and co-authors from these 2 areas, I think we can be very happy. But I understand that you will be very interested in comparing the Northern and Southern Benguela upwelling system. And I would also do this I also would love to do this, and maybe in a future project and a future effort, we can do this using this approach with stable isotopes and size spectra. I think this is a promising, let's say, approach and idea, very promising idea. But then I think we would need some other data and another, let's say, another sampling design, maybe with more cruises, standardized cruises in both upwelling systems.
Yeah.
But I think that you have such data and such samples, so I think we can talk about this later if you're interested.
Thank you. There is a comment in the chat in relation to what we are discussing. There is an observation that the difference in Northern and Southern Benguela in biomass would suggest a big difference in SST, that being Northern being cooler than SA. Is that correct, or is this correct?
I think that's a very important point. I think also that the temperature has a strong effect, maybe not so much on the predator-prey mass ratio and the trophic efficiency. In my view, these, let's say, feeding related and predation dynamics related parameters, they are more related to the food availability, that means to the density of food. But as you remember, there was another parameter here, which is the metabolic scaling. And the metabolic scaling, of course, has a very strong link to temperature. So these 0.75, which is given here as an estimate of metabolic scaling, has a huge variability too. There are many studies that are dedicated to estimating the metabolic scaling in different systems, and it varies a lot. And yes, it is related to temperature. So we must expect there to be a direct effect also of temperature on the ecosystems, and the size spectral slope is certainly also influenced by temperature. The question is the magnitude of these effects. Now, what is more important in explaining these data? Is it differences, for example, in oxygen concentration, in temperature, which would affect the metabolic scaling, or the availability of food and the density of food and the energy spent to search for food? But yes, this is an important aspect. Thank you for raising this.
Okay, well, thank you. That is from Els. Then there is another one from the same person. So for stable isotope results, the oxygen and carbon, we have to be patient of what kind of material will you measure stable isotopes.
For what kind of materials? Usually we measure the organic materials. That means we're not looking at carbonates. For example, as you say, the oxygen isotopes are often analyzed, for example, in foraminiferans using the carbonates. We are using only the organic fraction, and then for the larger organisms like fish and macrocrustaceans, usually we use muscle. For example, the fillet, the fillet from the fish, and the abdomen, muscle abdomen from larger crustaceans. And for the small organisms like zooplankton, we use the whole organism. And usually we remove the carbonates with acid before measuring. So this would be the way that we do the stable isotope analysis here in Recife. And I think these data that I presented, they were measured in France and in Brest. But here in Recife, we also do these measurements in our department. We also have a mass spectrometer.
Okay, thank you. Raise a hand from Kat. Kat?
Yes, thank you, Ralf. I always like talks that make me think. Now, first a technical question. Your abundance size graphs always show more or less equal abundance at very low size. Is that the measuring artifact, or does it mean anything?
Equal abundance at a low size. You mean equal abundance?
Well, you have no slope anymore at the very low size fractions.
Oh, you mean when at the end of the size spectrum for the large fish? You mean, I don't know whether you can see my mouse. You mean where we go for the large fish?
No, on the other end of the curve, at the lower end of the curve.
The lower end.
That's the one. Yes.
Here you mean here, for example, or here? Yeah,
where it says phytoplankton spectrum. Yeah.
Yeah, I think we still have some linearity here, but you are right. In this graph, we still have this linearity, but you're right. There is this debate about the dome-shaped size spectrum. No? And it doesn't show in this graph, but we have other graphs and other analysis where we debate whether, let's say, I don't know whether you can see my mouse, if you can see whether there's also a decline here in abundance in the lower organisms, and whether this is a real ecosystem phenomenon, or whether this is a artifact of the sampling, because we must remember that we cannot have a perfect representation of the ecosystem in our samples. We have, let's say, a lot of factors affecting the composition of our samples, and the most important one is undoubtedly the gear avoidance. No? That means that many organisms have developed strategies to avoid being eaten by larger organisms. And the most important one is maybe just by swimming away or jumping away, or some other strategies that would make them disappear in front of our nets. And of course, these strategies affect here the larger organisms, the larger fish. They are usually faster and can avoid our nets better, and also the larger zooplankton organisms, they can jump faster and have let's say, a jump that goes farther, so they can avoid our nets. But also for the small organisms, we also have gear selectivity effects, because the very small organisms, they will be able to slip through the meshes of the net. No, they are more likely to be able to escape through the mesh.
Well, I'm a microbiologist, and that's, as such, I'm interested in the green organisms there in your green part. I'm just wondering, how well can you resolve these very, very small sizes?
Usually for the phytoplankton, in the semi-automatic analysis, we use a flow cam, which is a little bit similar to a flow cytometer. That means you have a continuous flow of cells in front of a camera. And maybe your question is about the size of the cells, the minimum size of the cells that we can resolve. This will depend on the camera settings and on the size of the cuvette that we use. But this is also done with semi-automatic analysis with this equipment, a flow cam, that we also have here in Recife. We have a zooscan and a flow cam, and we have several colleagues who are dedicated to analyzing these unicellular organisms with a flow cam.
That's in the micrometer range.
Yes, in the micrometer range, but I would say in the larger micrometer range, I would say like between 10 micrometers and 50 micrometers or so range, I would guess. I'm not sure. I'm not a phytoplankton specialist, but I can look this up.
Thank you, Ralf.
Okay, thank you. I have one from my side. It's in relation to the uncertainty that you said in calculating the trophic level using the nitrogen isotope. I want to understand how your work in relation for trophic efficiency assists in reducing this uncertainty, especially the one that is related to the discrimination factors that is always assumed.
Exactly. This, you are completely right. The discrimination factor, which is, as you said, is very difficult to know precisely, which is the factor that is used to transform the stable isotope data to trophic levels. So these trophic levels here between 1.5 and 3, as I also said a few minutes ago, is very strongly affected by these factors. As you probably know, there is, when we talk about the difference in stable isotope ratio between trophic levels, there is the effect of fractionation, which has to do with the dynamics of the different isotopes regarding their mass and the transport through membranes and diffusion and the enzyme dynamics. This is the fractionation, which is very variable depending on the system. And we also have the routing, which is the way that the stable isotopes are distributed within the organism, no? And within the different kinds of molecules, like the different fatty acids and amino acids and so on. This is the routing. And the routing and the fractionation together, if we put these two effects together, is the discrimination, which you just mentioned. No? This discrimination is the difference in stable isotope ratio between prey and predator. This is the way that the stable isotopes change between, let's say, the French fries that I'm having for lunch and my own tissue in my own body. And this difference is then used to calculate the trophic level. It can be very variable and there's a lot of uncertainty. That's why we now together with colleagues at IOW in Rostock-Warnemünde, with a group of Maren Voss, we are also participating in a project with amino acid specific stable isotopes. So we get a more precise estimate of the trophic level. So nowadays, the most precise way to estimate the trophic level of an organism is using amino acid specific stable isotopes to measure the nitrogen isotopes in specific amino acids, where you separate the amino acids in a gas chromatography, and then you measure the N15 in the different amino acids. So this is, then you will have a very precise way to estimate the trophic level. But this is very, very time consuming. So you cannot do this for lots of samples. Like here, we usually work with hundreds of samples. When you do amino acid specific stable isotopes, you will just have, let's say, very few samples, like 10 samples or a dozen samples will be a lot of effort. So that's a problem here. But the combination of both methods, the bulk stable isotopes, like the ones that I presented, and the amino acid specific, if you combine this, then we are on a way to improve our estimates of trophic level and thus predator-prey size ratio. And thank you for raising also these questions about the discrimination factors.
Thank you.
Yeah.
Any other question I see, Kat? Is that an old head or a new one?
Well, I left the hand up because I do have another question, yes.
Okay, please go ahead.
Okay. There is one statement, Ralf, that makes me think even more. You had a sentence on one of the slides that says, oligotrophic systems are less productive. I agree with that, because they are limited by nutrients, right? They can't produce much. And then you said in the second part, and less efficient. Isn't that contradicting any ecological theory that oligotrophic environments select for the most efficient organisms?
Yes, that's a good point. And that's something that, let's say, the consequences for ecosystem theory have to be, let's say, evaluated. But from, let's say, a mechanistic perspective, from the ecosystem perspective, I think it makes sense to think that an oligotrophic system is a system where organisms are farther apart. You have larger traveling distances between patches of food, larger traveling distances between different prey items. So the predators will use more time and more energy to search for food. So if predators use more energy and more time, they will lose more energy, and thus this would make the system less efficient. The system would then be less efficient in the oligotrophic than in a system where you pack all the organisms very much together and it becomes very efficient. That's also, in my opinion, one of the reasons why the Peruvian upwelling system, where I have been working for many, many years, the Peruvian upwelling system is so productive because it's a shallow system. Even if you go to the offshore shelf, you have a water depth of 100 or 200 meters or so, but the oxygen minimum zone makes the productive area where actually everything is happening a very shallow. We have a very shallow upper layer, sometimes only 20 meters or 30 meters in the Peruvian system. And then you have all the productivity, all the organisms, the copepods, the anchovy, the phytoplankton, all packed together in this upper layer, and this makes the system very efficient. It's very easy for the predators to find their food. In an oligotrophic system, like in the tropical Atlantic, they have a hard time finding their food, and this makes the system less efficient. But what you just said is very interesting. Thank you so much for raising this, because of course oligotrophic systems will select for more efficient species and for more efficient let's say strategies, and this seems at first sight quite a contradiction, and this will be quite interesting to debate. No? Thank you so much for raising this.
Well, it boils down to what you mean with efficiency, but that's something to think about. Yeah?
Yes, I agree. Here it's about here's about the trophic efficiency of the system, and what you were just mentioning would be the selection for, let's say, more, for example, in the phytoplankton, more nutrient efficient strategies at individual level. But this is quite interesting to look at. Yes.
Thank you. Any other question or comment from any of the colleagues? I don't see any hand. Then let me just thank Ralf for the interesting presentation. I think we are also looking forward to the final result of the Triatlas project, which I think Margaret is also involved. And we are looking forward to the result.